Okay, so I'll talk to you today about our research at McGill here in the field of diabetes technology, with focus on type 1 diabetes. Is my audio fine? Yeah, for the Zoom. So several companies introduced continuous glucose monitoring systems in the last 10 years that measure sugar level in a continuous, non-invasive manner. And that really transformed the diabetes field into what we call now the diabetes technology field, where we start having new algorithms and new devices that we'll talk about that in the next hour. So the first thing I will talk about is automated insulin delivery system. So these systems are now commercially available and are composed of three components: uh, glucose glucose sensor that measures sugar level every 5 to 10 minutes, insulin pump that give insulin subcutaneously in a continuous manner, and the new component, which is a dosing algorithm that automate insulin delivery based on the sugar level readings. Now, just some terminologies. Yeah, so some people call them closed loop systems, some people call them artificial pancreas system, and there's one particular group in Harvard, they call them bionic pancreas, but these terminologies are used for the same devices. Now, in the last few years, there have been multiple devices that were commercially available, and they were all tested in randomized controlled trials, comparing what we call conventional sensor-augmented pump therapy. So this is people using a glucose sensor and insulin pump, but then they themselves decide on how much insulin they take, compared to automated insulin delivery. And what we see is that consistently, the percentage of time for which glucose levels spend in target range, and that is between 3.9 and 10 millimole per liter, is improved by around 11%. And that is an equivalent of 2.5 hours per day. So these systems now are commercially available. There have been studies looking at quality of life, quality of sleep, different studies in different populations. Just 2 days ago, they published in the New England a study in pregnancy. People they studied in 2 years old, basically in elderly people. And we see these systems over and over improve glucose control by around 2.5 hours per day. Now, clinically, what we see in the field, any improvement beyond 5%, we consider as clinically significant. So what is next? Now, if you look at the proportion of patients that spend time in range above 70%, which is the current guidelines recommendation, around 1/3 to 2/3 of the participants in these randomized controlled trials, they did not achieve glycemic targets. So despite doing much better than conventional therapies, we still have almost half the people do not achieve glycemic targets. And the other thing, all these systems, we call them hybrid closed loop systems, mean that they still require the patients to do what we call carbohydrate counting. So for every single meal they eat, they need to look at the meal, do the amount carb counting for the food, and then they enter the amount of carb in the system to get a prandial insulin dose that is proportional to the amount of carbohydrate they are eating. So basically what we're doing here is that we're trying to improve the systems by either improving the percentage of patients that achieve glycemic targets and simplifying or even alleviating the need of carbohydrate counting. So one of the questions we're trying to answer here at McGill, can adjunctive therapies improve glucose control with these systems? And can adjunctive therapies improve or simplify carbohydrate counting with closed loop systems? So we're looking at empagliflozin, an SGLT2 inhibitors, pramlintide, which is a drug approved in the US, but not here. It's an analog of the hormone amylin, and semaglutide. So just quickly about SGLT2 studies. So there have been multiple studies in type 1 diabetes, and the result has been quite consistent. So A1C reduction by around 0.4%, weight reduction, total insulin requirement reduction. But across all studies, we see an increased risk of DKA. Increased risk of DKA of around 3 to 5-fold. Now, the exception is the 2.5 milligram empagliflozin, which did not show any increase in the risk of DKA, but the glycemic benefit with that dose was actually more modest. So we recently did a study where we compared 25 milligram empagliflozin to placebo in people without using closed loop system. And the question we tried to answer, if you add 25 milligram empagliflozin, which is the highest dose of EMPA, to a closed loop therapy, how much improvement in glucose control you will actually get. So we did a randomized control trial where we had 4 weeks per intervention, 27 adults, average A1C was 7.7%. We compared 25 milligram EMPA with the closed loop system, 25 milligram EMPA with sensor augmented pump therapy, which means people are using insulin pump, a glucose sensor, but the insulin pump delivery is not automated. So they have to decide how much insulin to take. And placebo with closed loop system, and also placebo with sensor augmented pump therapy. So it was the system that we built here at McGill. So we use a tandem pump. We put our dosing algorithm on a phone, and we use a Dexcom sensor. And it was a two-side study. So us and basically Bruce Perkins and in Toronto. So these are the median glucose profiles. You can see clearly visually that if you add empagliflozin and closed loop system compared to sensor augmented therapy plus placebo, you have significant decrease in mean glucose level. Now, the increase in the percentage of time in target range was actually over 4 hours. So compared to closed loop system alone, that increased time in the range by around 10% or 11%, 2.5 hours. If you add EMPA 25 milligram on top of this, we actually increase the time in the range by around 17.5 percentage point, which is around 4 hours, a bit more than 4 hours per day. Now, one of the things we look at is ketone level. So the study was not long enough to look at DKA risk. So we looked at ketone level. So we were asking participants to measure ketone level every morning. Now, in this figure, what you see is the fasting ketone level on placebo. So again, I don't know why there is this picture here. Maybe let me. I can't when I. Maybe it's fine. We're afraid to touch it. Oh, okay. Okay, good. Now I don't see myself, but I don't care. You can see me. Okay, good. So, so the triangular, these are the sensor augmented pump therapy, and the circular are actually the closed loop therapy. And you can see on placebo, there is no difference really between the two therapies. When you go to lower BMI, you have a slightly more, or even more than slightly more, fasting ketone level with both systems. However, if you look at the data with empagliflozin, you can see that, well, first of all dots are more elevated compared to placebo for both sensor augmented pump therapy and closed loop, especially for people with lower BMI. But you can see, particularly with the closed loop system, the increase in ketone level was actually higher than sensor augmented pump therapy. And our interpretation, because the closed loop system often suspends insulin at night, so you have insulin suspension in addition to empagliflozin, and basically you have an increase in ketone level. So we did a second study with actually Melissa Pasco here. The question we tried to answer, does low dose empagliflozin as adjunct to closed loop therapy improve glucose control in suboptimally controlled adults with type 1 diabetes? So we define suboptimally controlled adults as people with A1C above 7%, so between 7 and 10.5%. And then we took these participants, we put them in a closed loop system for 2 weeks, and we confirmed that their time in range was below 7%, 70%. So again, these people, their A1C is above target, and despite using closed loop system, which is the best available insulin-intensive therapy, the proportion of time their sugar level in target range was still below 70%. So we really took people who still cannot achieve glycemic targets. And then we recruited 24 adults, and the average A1C was 8.1%. And we wanted to see whether the low dose empagliflozin improved glucose control. Now, again, in the previous studies, we showed no increased risk of DKA with the low dose, but the improvement in time in range or even in A1C was actually quite marginal. So we did a randomized control trial where we had 2 weeks per intervention, where we compared EMPA 2.5 milligram with closed loop system versus EMPA 5 milligram with closed loop system and versus placebo with the closed loop system. Now, the time in range for people in placebo was around 59%. So again, the average proportion of time people spend in target range is almost basically 12 hours, almost 15 hours per day. Now, with the 2.5 milligram, we actually went all the way to 71.6%. And with the 5 milligram, we went to 70% in time in range. And you can see that in this population, the improvement in time in range was quite significant. And importantly, when we measured mean capillary ketone level in the morning, there was actually no difference between the three arms. Now, when you look at the two studies together, the improvement in time in range with the 2.5 milligram EMPA was actually higher than what we have seen with the 25 milligram EMPA. And the reason, because of the lack of the ceiling effect. Like the other study, participants had good glucose control at baseline, and there is less room for improvement. While in this particular study, we took people who still have more room for improvement in their control, and you give them even a low dose of empagliflozin, we actually saw a significant improvement. And again, this improvement is around 11%. So basically, what you're saying is that by giving people low dose EMPA, you double the benefit that you can see with closed loop system. Now, this was only a 2-week study. So the question we are now trying to ask, can we confirm these findings in longer trial, a multicenter trial? So we're starting soon a study in a parallel design, multicenter, so 3 sites. So where the lead site at McGill, a second site at RCM here in Montreal, and a third site in Toronto. 46 participants, where we're comparing 2.5 milligram EMPA on people on a commercial closed loop system versus placebo. And again, we're taking people who are using commercial advanced closed loop system, and their time in range remain below 70%. So we're taking people who still struggle to achieve glycemic targets, and we're randomizing them to 2.5 versus placebo over a 6 months period. So this study is multi-center, much longer, and looking at people using commercial system to try to basically confirm the findings of the previous study. And this study is really, I mean, it has some clinical implication because you have people coming to clinic who are using commercial closed loop system. Their time in range is 50-55%. Can you actually give them low dose EMPA safely and improve the glycemic control? And this is the question we're trying to answer in this particular study. Now, the other thing we're looking in that space, so recently there's actually a Chinese company that is selling continuous ketone monitoring system, and Abbott is developing a dual continuous glucose and ketone monitoring system. So we have a study that we're starting soon to use this ketone system in people with type 1 diabetes using very low carb diet or a long intermittent fasting. And then we're planning to do other studies with people using SGLT2 inhibitors. And the idea is that if you're using continuous ketone monitoring system, you can use higher doses of SGLT2 inhibitor in type 1 diabetes to get better glycemic outcomes, but also better renal and cardiac outcomes. Because if you look at the studies with different doses, we seem to see glycemic outcomes saturate quite early, but renal outcomes, the more SGLT2 inhibitors you give, the better outcomes actually you get. But we cannot use it safely in type 1 diabetes. And the question we're trying to see is whether we can use it safely if someone is using a continuous ketone monitoring system. So the second drug we're looking at is pramlintide. Maybe more people are less familiar with this one here. It's basically an analog. Oh, what did I do? Okay, it's an analog of the hormone amylin, which is co-secreted with insulin in people without diabetes, but completely lost in people with diabetes. So this data from, I believe, 8 healthy individuals, and you look at the plasma insulin, plasma amylin, they actually follow the same profile during the day. And again, if you have type 1 diabetes, you lose the ability to secrete both insulin and amylin. You get insulin replacement therapy, but you don't get amylin replacement therapy. It's completely lost in type 1 diabetes. Now, the main action of amylin, a bit similar to GLP. So basically, slow gastric emptying and suppresses the glucagon secretion and induces satiety. Now, this study, if you look at the glucagon, so this is postprandial control in people without diabetes. After a meal, you can see their glucagon level is quite constant postprandially. However, in people with type 1 diabetes, in this particular study, they show an abnormal increase in the glucagon level after a meal. Now, in the second study, you can see that the placebo, which is basically the white boxes, people eating a meal with type 1 diabetes, and again, you see this abnormal increase in glucagon level, but when people are taking different doses of pramlintide, their postprandial glucagon level is more normalized and looks a bit more like people without type 1, without diabetes, basically. So what we're doing in the lab, we build a system which is insulin and pramlintide closed loop system that instead of delivering only insulin, we also deliver pramlintide. Now, the idea is to combine the two in a co-formulation. Now, we don't have co-formulation, so we're using 2 insulin pumps. One we put insulin, the other we put a glucagon, and we run different kinds of studies to see how much improvement we get if we are using both hormones. So I just talk about a couple of studies here. So one study, we did 2 weeks per intervention, 15 adults, 15 pediatrics, where we compared fiasp and pramlintide with meal announcement. Now, meal announcement means at meal time, instead of doing carbohydrate counting, you take your phone, you press out a button, and that's it. The system will give you a small insulin priming dose, and the rest will be based on your sugar levels. And then the second arm was FIASP and placebo with meal announcement. And the third arm was conventional commercial closed-loop system, where people have FIASP and placebo with carbohydrate counting. Now, this is the graph. So the blue line is basically the control, which is FIASP and placebo with carb counting. Now, the green line is with placebo. Basically, what we see here is that if you're telling people don't do carbohydrate counting, without giving them pramlintide, you have degradation and glucose control because they're not getting enough prandial doses. However, with pramlintide, despite people not doing carbohydrate counting, we still increase the percentage of time for which glucose levels in target range by around 5%, so 1 hour per day. So again, here what we're doing, we're alleviating the burden of carbohydrate counting and at the same time improving glycemic outcome. So what we did actually interviews. So we did interviews and we did qualitative analysis. So here I'm showing you some quotes from the patients because again, these people are used to do carbohydrate counting and suddenly we're telling them, go on for 2 weeks, don't do carb counting, and we will look at psychosocial outcomes. So this is from an adolescent. He or she said it made it easier with not having to count and the eating. And it made it like less complicated and more like, oh, I'm hungry, I can eat. I don't need to like count stuff, which was nice. And the second participant said, in adults, I found it was more a natural way to operate, but also not having to do that calculation. It still removed the weight that is so usual that we don't realize it made a big difference. Another adolescent said it avoided having my parents tell me it's my fault because I'm counting my carbs wrong. Another quote that people felt it restored more freedom and normalcy. This participant said, adolescents again, if I had a meal and I thought it was too big and I gave myself insulin, I didn't have to finish it. So that gave me more freedom. So again, because with conventional therapy, if you look at a meal and say, well, this is 100 grams of carb, I'm hungry, I will eat it all. You give 10 units of insulin, you really need to eat 100 grams, otherwise you will have a hypo. So again, with the system, people don't need to feel like they can start eating and they stop whenever they feel like stopping. Now, one actually interesting comment that came more on the negative side, I felt like the meal announcement strategy offered me liberty that didn't necessarily benefit my health. So people don't do any more carb counting and more conscious about their eat. So that was an interesting comment that came from this participant. Now, the other thing we try to do is pramlintide to do fully closed loop. So fully closed loop means you wear the system and you don't even do any announcement at mealtime. And we have an algorithm that look at the sugar level, and based on the sugar level reading, we can actually detect the meals automatically 30 to 50 minutes after the meal, and we start to give a small insulin and pramlintide boluses. So this is one of our trials here. So you can see the red arrows, these are carbs of carbohydrate. So the lunch was 96 grams, so it's already quite a big meal. And then the boluses are coming after the meal. So the system doesn't know about the meal, look at the sugar level. When they start increasing, detects the meal and start to give a small insulin and pramlintide bolus. So we did an inpatient trial where we compared basically a fully automated FIASP and pramlintide system. And we used 1:10 ratio, the delivery between the two hormones. Versus again, the control arm was comparable to the commercial systems, FIASP alone with carbohydrate counting. And people came 24 hours to clinic actually to hear at the sim, and they had basically standardized snack, breakfast, lunch, and dinner, and they did some exercise. We had 24 adults, and A1C was 8.1, and these are the outcomes. Now, if you see the first graph, which is the sugar level, you can see with the fully closed-loop system, which is the blue line, we can have some, degradation in glucose control in the early postprandial period. And the second banners where insulin delivery are, the red boluses are coming at mealtime. This is when you had basically carbohydrate counting. But the blue ones, this is the insulin that is being delivered with the fully closed loop system. And we're basically replacing pre-prandial insulin doses with post-prandial insulin and pramlintide doses. Now, if you look at the numbers, the time spent in target range with the fully closed loop system was 74%. However, with the hybrid closed loop system was 78%. So we have some degradation in glucose control, but again, these people had to do nothing with the system. They just wear the system and they do absolutely nothing at mealtime. And now we're actually moving toward outpatient studies where people wear the system for 3 weeks, and we look at both glycemic outcomes as well as psychosocial outcomes. Now, our studies are really useless until someone develop an insulin and pulmonitide co-formulation, because no one will be using, or maybe very few people are willing to use two pumps. But there are multiple companies looking at formulating both hormones in a single drug. And Adocia is a more advanced one. It's a company in France, and actually we did for them here a first-in-human clinical trial with one of their co-formulations. So Adocia in France, Ecor in England, and basically there's a group in Stanford in the US looking at co-formulation. And there are all kinds of questions: what the ratio between the two hormones, which insulin to use, and people are looking at all these questions. So the other study I want to talk about is semaglutide, so Ozempic. So we have an ongoing trial. So actually, Melissa is running the study, and Michael Sukas. And basically, we're having 11 weeks run-in on semaglutide, and then we have 4 weeks on insulin alone hybrid closed-loop system. And the question we're trying to answer is that if someone using closed-loop system, how much glycemic benefits you get by giving them semaglutide? And you can't just take studies from type 2 and assume they work in type 1, because insulin secretion is completely lost in type 1 diabetes. And we do the same with placebo. And this is, to my knowledge, I mean, Melissa finished recruitment, and the last participant should finish in April. And this will be the first trial in type 1 diabetes using semaglutide. Now, the other study, and we do with semaglutide, we do titration till 1 milligram or maximum tolerated dose. So if people can't get to the 1 milligram, we actually stop a bit earlier. So still ongoing trial. Hopefully we get the results back in April. Now, the other study actually that Michael Sukas is leading is looking at Longev and Ozempic. But this time, we're not looking at the improvement in glycemic control, but alleviating the need of carb counting. So we have 11-week run-in on semaglutide, and then we have 4 weeks on a commercial closed-loop system with full carbohydrate counting, 4 weeks with simple meal announcement, and 4 weeks with fully closed-loop system, where people wear the system and don't do anything around meal. And then we do crossover with placebo. And the question here we're trying to answer, if someone is wearing a commercial closed-loop system, can I give them semaglutide and ask them to reduce basically the effort they have to do around carb counting and around basically meal intake? So that we still haven't started. We're now writing the protocol, but it's actually funded. Now, the other thing we're looking at more a bit far from the drug side is that can we actually just simply with insulin replace carbohydrate counting with what we call a qualitative meal size categorization? So people at mealtime, instead of having to decide if there's 45 gram or 47 or 53, they just say, okay, this is low carb, medium carb, high carb, or very high carbohydrate. So we run a study with RCM to look at is the qualitative meal size categorization non-inferior to carbohydrate counting with insulin alone. So again, no adjunctive therapies. So we had a randomized control trial, crossover design, 3 weeks per intervention, and we had 2 sites and 30 adults with type 1 diabetes. And average A1C is 7.4%, so it's more better control than the previous studies. Now, the carbohydrate counting arm, the time range was 74%. Now, with the qualitative meal size estimation, we dropped around 71%. So again, you can see this from either say, well, time in range with the meal size categorization was actually great, 71% on average, but also we had a slight degradation in glucose control when you don't ask people to do an accurate carbohydrate counting. Now, we didn't do a formal qualitative analysis and interviews, but talking to patients, like overwhelmingly, they were happier with qualitative meal size estimation, because 3% is 45 minutes a day. And unless you're actually looking at your sensor and counting your CGM values, you really don't feel it. So people are really much happier when you alleviated the need of carbohydrate counting. And we should start study soon with Longev. Actually, Caroline here, she was doing that study. When we're comparing carbohydrate counting with the qualitative meal size estimation, was fully closed loop. And for that study, we're using Longev. So it's faster insulin. And the idea with faster insulin, you can be more aggressive with insulin delivery postprandially. And we're using the Ipsomed pumps. It's a new system we built here based on a Swiss pump called Ipsomed. And we should start next month, hopefully, yeah. So the other thing I want to talk about is multiple daily insulin injections. Now, the idea is that people who use injections, if they're using glucose sensor, you can't change their insulin dose every 5 or 10 minutes. But what you could do, you could actually build apps that look at their sugar level over the previous week and give them automated recommendations to how much insulin they should take for the following week. So again, we're not changing delivery every 10 minutes, but actually we're providing more a decision support system. So people call it open loop algorithms, people call it decision support systems. Basically, they're all the same. So basically, we're given glucose levels, insulin injections, carbohydrate intake. For example, this is data from a CAMP study where we have the sugar level, we have the basal dose, we have the carb intake. And the question is, here this patient have hyperglycemia in the night, they have hypoglycemia after every meal. We need to make an adjustment to the basal dose, the carbohydrate ratio, or the fixed meal doses, and the correction factor. And can we improve glucose control? So we built algorithms, and we did study as the first in a diabetes camp, it's Camp Carowanis in Santagat, where we did a non-inferiority trial where we compared 11 participants, or kids actually, where their parameters were being changed by the physicians of the camp daily, versus another 10 where the algorithm was making the changes daily. Again, it's a pilot study. And here what you can see, each color represents a different kid, and each column represents a different parameter. So basal dose, breakfast carb ratio, lunch, and dinner. And you can see that both the physician and the algorithm were changing significantly the parameters of the patients. And you can see also, if you look at the average change over a day, from the beginning of the camp till the end, it was converging. So at the beginning, we did more changes, and toward the end of the camp, the algorithm and the physician were making less and less changes to the parameters. Now, before we actually implemented the algorithm recommendation every day, the physician of the camp, we had actually 6 different physicians, every week different physician, had to approve it. So we have the approval rate of around 92%. And this agreement between algorithm and physician was around 2%, 2.5%. So this agreement means the physician say increase the basal, the algorithm actually want to decrease the basal. Now, the glycemic outcomes were similar. But again, this was not properly powered. So we cannot do p-values and see whether the differences were actually statistically significant or not. Now, then we built actually an app that we call the iBolus app, where it's a bolus calculator, but runs on a phone. So patients can have this phone, and at mealtime, they open the app, they put the amount of carb, they choose the meal, and the phone will actually give them the amount of insulin they should be taking. And the idea is that we can actually update the parameters in the background of the app to make the app actually smarter over time. And every week, we'll actually learn how much insulin this patient needs. And we built a platform so we can actually log in and we can see the sugar level of the patient while they're using the app at home. This is data over a week. You can see the boluses, the missed basal. You can see different alarms, and you can get a feeling about what's happening on our end here. So we built that here at McGill. And then we ran a randomized control trial in 84 adults. We just finished recruitment actually last week, where we recruited people with baseline A1C above 7.5. And we compared over 3 months what we call sensor-augmented multiple daily injection therapy with an app. So this is the control arm. So these people are using multiple daily injections. They have the app, which is just a basic bolus calculator, and they have a sensor. And that's it. Now, the app doesn't learn over time. So we go in the app, you program their fixed boluses or the carb ratios, and basically the app does the calculation automatically for them, but there is low learning. And the other arm, we did a weekly learning. So every week, the app actually automatically will receive data from the cloud to adjust their basal dose and the insulin to carbohydrate ratio. And the primary outcome is looking at A1C. And the idea is that with sensor-augmented pump therapy, people can still change these parameters. So, they see the physician one every 6 months, they change the parameters. But by making these adjustments regularly, we can improve A1C. So this is just a couple of patients. You can see this particular patient, at the beginning, their basal dose was, 18 units, and we start increasing that gradually to around 26 units. And at the same time, we decreased their carb ratio, so we're giving them more and more prandial insulin. And you can see their time in range from week to weeks, over 12 weeks, actually improve over time. So another patient here where the algorithm reduced the basal dose from 75 units all the way to around 55. So this is quite a big reduction in the basal dose. Again, every week, so it's a gradual decrease in the basal dose. And at the same time, interestingly, the algorithm was giving this patient more and more prandial insulin. So that means this patient probably having hypos at night, but their daytime control is good. So the algorithm reduced the basal to prevent the hypos at night, but then you get less insulin during the day, and we start giving you more prandial insulin to compensate for that. And if you look at the percentage of time in hypoglycemia, so the lower figure, it drops significantly from 10% all the way around to less than basically 2 or 3% and remains there. Now, again, these are two sample patients. Not everyone works very nicely like this. So study is still ongoing. We should finish in December. Now, however, we also did interviews. These are some quotes from some of the patients. So this patient said, just referring to the algorithm, I was actually surprised. The fast-acting recommendation, so he meant or she meant the prandial doses, were pretty much on par with what I anticipated. But what I was surprised with was the long-acting insulin recommendation. We dropped the bather significantly, which has eliminated my concern for having low sugars overnight, and still has maintained a steady level throughout my actual working time of the day. Another patient said, the 6 months patient visit in the hospital, to let that go for such a long period of time could have negative consequences to one's health. This at least, a guidance immediately in real time. And again, it does give the patient the option to override. Changing the doses is something we rely on medical professionals to do. I've worked on changing them by myself at times, and there's always that fear, have I gone too far? So if the application is as safe as professional, then it's like having that contact on a regular basis. Now, some people commented on the app itself, so not about the algorithm. So this is the control arm. I like the fact that it kept track of how much insulin I had in my body at the time, so it won't overdose me, which cannot do on my own. So we're actually showing them the insulin on board all the time on the app, so they know how much insulin still not fully observed, but actually has been delivered. So even that on its own, on the control arm, people find quite helpful. Now, the other study we're doing is how does the algorithm compare to a group of endocrinologists? So we actually took data from the study, 12 participants, and then we took this data over 4 weeks, and we actually gave them to 16 endocrinologists here at McGill. So Simon is one of them. And every week they go in, they look at the data, and they say, well, how much they think the new recommendation should be. So again, this is a retrospective analysis. We don't know whether the physician recommendation actually works or not, but we just took their recommendation, and then we compared that to the algorithm. Now, a bit later, we gave the same data to the physician to look at the variability within the same physician. And then we did the same, but giving them a 2 weeks data. So again, we just wanted to see, can we claim the algorithm is one of the physicians? Is it performing similar to the others? Now, this is the first time I show the data. So the full agreement. So full agreement means one of the physicians increased the basal, the other one increases the basal. So we're looking at full agreement in the direction of change. So among physicians and the basal rate around 50%. So that means only 50% of the time, two physicians actually increased the basal rate or decreased the basal rate when we did the pairwise comparison. And the same for all the carb ratios. And the algorithm was also around that, around 50% of the time, the algorithm compared to the other physicians. Now, what we call partial agreement. So partial agreement means one physician increased the basal and the other physician doesn't change the basal. And also that was around 40, 45%. And again, when you compare the physician among themselves and the algorithm across the physician, we get similar numbers. Now, interestingly, what we call full disagreement is around 5 to 10%. Again, among the physicians and between the algorithm and the physicians. So again, this means around 8 or 10% of the time, one physician increase the basal, the other physician actually decrease the basal. So that gives you that is really quite subjective manner of these changes. And there's a really large variability between physicians. And I think, again, we have to do p-values and so on. But again, looking at this, looks like the algorithm is not far from the physician's recommendation. Now, the other study we're doing is what we do. We have an app, and the same app actually added high-fat meal. So when people eat high-fat meal, they press they're eating high-fat meal, and the algorithm or the app recommends splitting the boluses, and then it personalizes that split over time. So they give some bolus at mealtime, another bolus 2 hours later. And also we do the same for exercise. So when they exercise, they announce the exercise to the app, and the app recommends reduction of the bolus. And also we improve that over time. Now I'll be quick. I need to make sure there are enough time for a question. So again, we did a single arm trial, 16 patients. And basically what we see is that the time below actually dropped for postprandial exercise. So whenever we had exercise, we looked at the data around that time, and we dropped the hypoglycemia from 5.3% for the first month till 1.4% at the last month. Again, this is a non-controlled trial, so single arm study. And what we find interestingly that there is large variability between participants and there's large variability between different type of exercises, which really basically emphasizes the need of personalized recommendation, where you cannot just tell everyone decrease your bolus by 30%. It really depends on the individual and the exercise they're doing. Now, this is maybe not very scientific, but just to give an idea about the field. So in the last basically 10 years, these are all companies that came to the market and products that came to the market. So the one on the left, these are technologies for people that are using multiple daily injections. So either what we call a smart pen, so a pen that track insulin doses that actually communicate to the phone, or a cap. So a cap is a small cap. Basically, you put it on insulin pen and track your insulin. And on the right side, these are all insulin pumps that actually recently came to the market to develop basically a closed-loop system. So again, you can see the development of continuous glucose sensor really opened the field for all these companies to come. And most of these are smaller companies, and some of them got acquired by larger companies. Now, the other thing, we had apps. So Camp, the app is basically an app from Cambridge that talks to insulin pumps, the closed-loop system. There's DreamMed from Israel. There's Tidepool. I'll talk about it more in a bit, and the Class AP, which is actually from us. And then there's also some innovation happening on the drug side to actually support the field. So Zealand and the rest are developing a glucagon that is stable in pump to be used in a dual hormone system that give you both insulin and glucagon. Arecor is doing basically the insulin, well, Arecor actually doing the insulin pramlintide, but also doing a concentrated insulin. So you can use 500 and the same PK as usually U100, and then you can use smaller devices. So there's a lot of innovation happening on the drug side as well that is driven by diabetes technologies. Now, the last thing I want to talk about is something called We Are Not Waiting. So there's actually a group of patients and family and friends of patients that around 10 years ago decided they're not waiting for pump companies to bring these devices to the market. So they went ahead and actually hacked insulin pumps and a glucose sensor, and they built closed-loop systems around 5 years before the first commercially available closed-loop system came to the market. And that gives you basically an idea about how much these patients and their family really needed these and wanted these devices. Now, that actually software that came from the community led to a company called Tidepool, and they just got FDA approval in January. So that actually innovation, was community-led innovation, made it to a company that now it actually is FDA approved to be used in the US. So again, this is purely driven by patient and community of patients. Now, one thing we do here, what we call community engagement ourselves. So this is diabetes camp. So we have actually diabetes camp here in Santa Gat, actually started by a pediatrician at McGill 60 years ago. And we do studies there. It's easier for us. We go there, we spend 6 weeks. We do a study that will take us 2 years to do at the hospital. Now, these are the tents. I've heard these tents haven't changed in a long time. And this is the map of the camp. And each dot is actually a tent. And it's around, from the first tent to the last tent, around 300 meters, but it's really a lot of hills. And when we were doing the studies there, I would do around myself like 1 in the morning. It takes me around 90 minutes to go over each tent. I need to make sure, our device is working and connections are good and so on and so forth. And then I felt, okay, maybe, these kids, a lot of them are having hypoglycemia, like not part of our studies. And we need to find a way to actually detect this hypoglycemia in real time during the camp without having to go over each tent. So we took a device called Meow Meow, which is a device that you put on top of Freestyle 1, which is the previous Freestyle, which was not real time. So when you put that device on top of Freestyle 1, it changed it into a real-time sensor. And we had 3 engineering students at McGill, they're undergrad students, and they actually took the codes that were developed by the We Are Not Waiting community, and then they took that code, they modified it, they put it on a phone, and we put a phone under each bed. So these are the tents again. So each bed, we actually put a phone under each bed, and then we cover the entire area with Wi-Fi. And over there, even phone signal actually doesn't work. So when we were doing our study, we used actually walkie-talkie to hear ourselves in the camp. So cover the entire area with Wi-Fi, and then we got all the data automatically to the infirmary, which is in the middle, and we donated to them big screen, and they were able to see the sugar level of each kid in real time with all kinds of alarms and different colors for different, low sugar level, high sugar level. So this was quite nice, which we did for the camp. This was before, like now you can do it with commercial systems. You can use the Dexcom share feature, but this was actually 4 years before that feature came. And the camp used it for 3 years where they could just, instead of waiting for the hypothete in the morning, they could go and treat it before it happens. Now, the last thing we did two Cafe Scientific, where we invite around 80 people with type 1 diabetes and their family, and we talk about our research. So this is actually poster from, we had someone illustrator where she was actually, as we present our studies and about the field, she was actually drawing the presentation, and this is one of the outcomes from one of the Cafe Scientific we did here. So we try to do research, but also try to engage with the local community, either through working with the camp or just through educational activities. And I have 5 minutes for questions. Thank you very much.
Thank you so much. Wonderful. Really interesting to see this. Congratulations on the innovation and for advancing this field. Really impressive. And by 6 weeks in a summer camp, that's quite a commitment there. Congratulations on doing that.
Yeah, not me. The whole team.
The whole team. Fantastic.
We go there, we sleep there. Yeah.
That's fantastic. I'm trying to see the questions. While I'm waiting for the questions to come in, tell me something about, the time spent in range. You told us a lot about time spent in range and comparing different percentages. What is just this big picture, the clinical outcomes? What percentage does one want to spend in range, and how does that change the clinical picture, big picture?
Yeah, so the current guideline is that you try to aim for a time in range above 70%. And people claim if your time in range above 70% on average, your A1C would be below 7%. Now, you cannot, the DCCT trial showed that if you are below 7% A1C or even below 6.5%, then you actually reduce significantly long-term micro and macrovascular complications. But we don't have similar data with glucose sensors. So people started looking at correlation between what is time in range that correlates with what A1C. And currently, the consensus is that anything above 70% should give you an A1C on average below 7%. And this is our guide. This is our basically target. Yeah.
Thank you very much. Are there any questions in the audience?
And the time in range between 3.9 and 10 millimole per liter. Now, the pregnancy time in range is different, and the peace time, but in general, it's 3.9 to 10 millimole per liter. And the time below range in hypoglycemia, the current recommendation is below 4%. So pump and a sensor, the closed loop system currently are the best insulin intensive therapy, basically. Yeah. But they're still not perfect. Yeah. Yeah. So the question is if I could comment on pancreatic transplantation. So this is really not my expertise. But when I look at some of the glucose sensor outcomes for some of the people who did the transplantation, we can never beat them with technology. Their time in range is 95%. They're more like non-diabetes people. But my understanding, again, it's not my field. There are a lot of other factors preventing people from using it. I think you have to go immunosuppressive therapy. I think there is cost. My understanding is that it's not easily accessible compared to closed-loop system. Plus, I'm not sure how much you can actually sustain these benefits. But definitely, we don't get there. They get better control than we can get with this technology, way better control. Yeah. Yeah. So the question is that are there any people who do our studies and other studies are more motivated people? So is there any data in the general population? So actually, the large studies that were done, the FDA requested from each company to recruit a minimum number in different A1C brackets. Now, interestingly, it's different what you would expect. When companies start to publish real-world evidence, their glycemic outcomes were way better than the outcomes you have seen in RCT, because not everyone who is doing the RCT can actually afford to get these pumps. So if you talk to companies, I don't want to start naming companies, they start to claim, oh, people get 80% time in range in our system. Well, no, these people who can afford your system get 80% time in range. But if you look at your own RCT, your time in range was actually 68%. So, yeah, yeah.
There's 1 minute for one final question. Can you comment on glucose variability determined by CGM and how you deal with this?
Yeah, the glucose variability. So people who do studies with the closed loop system, they look at the CV as one of the outcomes. Some studies showed reduction, but not all studies. We see more reduction with the standard deviation. And the reason is that the closed loop system reduces the mean glucose and the standard deviation together. So if you look at the CV, basically you don't see much difference, but the standard deviation is actually being reduced in most of these studies, especially at night.
Thank you so much. We're up on the hour. It's 1 o'clock. Thank you, everyone, for being here in person, and those of you online. Thanks for joining us. Congratulations. Terrific. Continue. This is great to see some innovation like this going on at McGill. Thank you so much for your contribution.